So thank you very much for the introduction, and it's really a pleasure and honor to be able to speak to you about our proposed research on using synthetic biology to produce cannabinoid molecules from microbial sources. And this is as Andre mentioned, this is a joint grant between myself and Professor Neil Garg from the chemistry and biochemistry, as well as in chemical engineering departments. So just first, since this is quite a departure from the last talk, I want to quickly bring everybody up to speed on what synthetic biology means. This phrase has different meanings to many people, but in the context of this talk, I listed two general definitions. I don't need you guys to read it, but I'll just mention that synthetic biology, the way I see it, is the use of molecular biology tools and techniques to forward engineer cellular behavior, or to combining the design and construction of enzymes, pathways that can be towards a particular purpose. So there is significant global impact of synthetic biology in recent years. The one notable example is the $15 billion Ginkgo Bioworks public offering just this month. And they took the symbol DNA, which is used to be a Genentech symbol. So, and their model is to grow everything and produce everything from engineered cells. Well, we are working towards that here in our lab. My lab focuses on small molecule bioactive compounds known as natural products. And many, if not all of us, have taken natural products as drugs or nutraceuticals, and they come from bacteria, fungi, and plants. And some of the more famous ones are shown here, and I'm sure this audience are very familiar with these drugs. The focus of our this research is on what's so-called the psychoactive natural products, and this picture was taken from a recent review that we wrote in 2021, and you can see many of the natural products produced by plant and as well as fungi here that have psychoactive bioactivities. And there has been a significant approach, synthetic biology efforts in producing these compounds from more sustainable sources, such as microbes, instead of relying on the harvest of these compounds from plants. And I'll get into that, why that is in a second. And one notable example in recent years is the example of making opioids from yeast. As we know that opioids are traditionally produced from poppy, opium poppy, and it takes a long time to scale up, to grow the plant, and to harvest the opioids. But recent progress from researchers at Stanford and other places have allowed the production of a very complex opium molecule from yeast cells by combining genes from different sources. And that can significantly lower the costs and the duration, and also making analogs of opioids that are not addictive. So let me give a brief overview of the molecules of interest for this talk. We're interested in the cannabinoids. These compounds are, of course, do not require too much introduction, but very briefly, I'll mention that these are natural products, psychoactive natural products produced from different cultivars of cannabis plants, including indica and sativa. The structures of the active components of these plants, recreational plants, were elucidated in the 1960s, and are shown here as cannabidiol, CBD, and tetrahydrocannabinol, THC. They were found to modulate the endocannabinoid system and hence have significant effect on human neurobiology, if you will. And in recent years, in the last 10 years, they have been promised as drugs that can treat a number of diseases. So these compounds affect the endocannabinoid system, which are named after these compounds, through interaction with the receptor CB1 and CB2, as well as other receptors. And the beneficial effects include decreased nausea, lowered feeling of pain, antidepressant, etc. And both CBD and THC have been clinically approved as purified compounds to treat a variety of diseases. And there's also people have proposed that modulating this endocannabinoid system using compounds such as cannabinoid may have therapeutic potential in almost all diseases affecting humans. And a conservative projection market for these compounds in purified form for medical use is to exceed $10 billion by 2025. So traditionally, these compounds have always been isolated from plants and still are. While growing cannabinoid plant is definitely a very attractive way of producing these cannabinoids and for other purposes, there are significant disadvantages if one wants to scale them up or for as a source of purified compounds, because they're quite expensive to grow. There's significant land requirement. These compounds are produced as minor components of a complex metabolic pool. It's present in complicated mixture, requires purification. Plants are relatively slow to grow, and it can be inconsistent depending on the weather or other factors. And there's significant regulatory burden of producing these compounds from plants. And if we compare that to a microbial production, if one is able to do so, we lower the cost significantly, and it's easy to scale because these can be fermented. Equipment requirement is simple. And we can engineer these compounds to be produced at high levels, so getting pure compounds is relatively easier. And because the nature of the genetic nature of the microbes, they're easy to engineer, and the pathways can be changed to make analogs. And because it's in a controlled environment, it's consistent, scalable, and it can be argued that there's less regulation to produce these compounds from microbial sources, although we are going through the regulatory process now to obtain approval, and doesn't seem that easy at this point. So how do we produce cannabinoids? Here's a chemical view of how cannabinoids are produced. Very briefly, these compounds are made by a set of 4 enzymes. Each compound is made by 4 enzymes. They start with olivatoic acid synthase and followed by olivatoic acid cyclase. These 2 enzymes will make a key intermediate called olivatoic acid, and also chemically it's been labeled known as a beta rososilic acid. And this compound is then prenulated by a geranyl diphosphate using a prenyl transferase to make the compound cannabidiolic acid, CBGA, which itself is a very high value compound, and it's being pursued by numerous synthetic biology companies, including Ginkgo, the company I mentioned earlier. And then this CBGA then is cyclized by different cyclases into either THCA or CBDA. And then upon heating, they will decarboxylate to release the pure components, active components, THC and CBD. So this pathway has been explored for synthetic biology applications. And most notably, Jay Kiesling in 2019 published a paper in Nature reconstituting this pathway in yeast. However, the yield, as you can see on the figure below here, is quite low in the milligram per liter level. And our own Jim Bowie in chemistry and biochemistry developed a cell-free platform to perform prenylation and production of CBGA. And using his optimized cell-free system, they can reach pretty high concentration, albeit the relative volume is much smaller compared to what fermentation can afford. Therefore, we like to tackle this project, this production of cannabinoids in this project. And here I listed the challenges that we saw and the goals of our project. The first challenge, as I mentioned, is that the current approaches for microbial production of these cannabinoids the titers are relatively low because these plant enzymes are relatively poorly expressed and not as functional when removed from their natural hosts. So our goal in this project is to develop alternative microbial hosts that can produce these compounds at much high levels that can lead to commercialization. The second challenge is IP limitations. The pathway I showed you on the previous slide is the only known pathway to olivatoic acid, these plant enzymes, the key intermediate, and the IP is heavily protected. So for anybody who wants to enter the space, we need to find a completely new pathway to this intermediate and analogs that can result in new IP for entry. And the third is diversification. Of course, the CBD and THC can be obtained from these plants, but as I'll show you in a slide later, analogs of these compounds with slight variations in the side chain structures have much better activities, are known to have much better activities. However, they're much more rare to recover from plant sources. So our goal here is to use our system and use the biofoundry facilities to develop a flexible synthetic biology platform to making different analogs of cannabinoids. I'd just like to show you our thinking really quick is how do you find an enzyme, a pathway that can get you this compound, alpha-letoic acid. So we've done quite a bit of preliminary data, and we focus on a related pathway that we've studied in my lab for a long time, which is these pathways that make these residuocylic acid lactones themselves are very interesting natural products that have diverse biological activities. What you see here are two large enzymes called polyketide synthase, which I'm not going to go into detail, that one of them makes a reduced compound showing red, and this compound then is fed into the second one, makes a non-reducing version compound, the 2,4-dihydroxybenzoate moiety, and then followed by macrocyclization to yield the structure shown here. And if you compare this structure to alpha-letoic acid, you see that we have the core component maintained, while this portion is different. So we thought that we can utilize the number of fungal genomes that have been sequenced in nature and find pathways that do not cyclize in this macrolactone format and produce chains, this red can produce parts of molecule that this red chain mimics more closely to that of alpha-letoic acid and related cannabinoid precursors. And so this is what we did, and we found a number of pathways in fungal species from just public databases that contain this feature, and we had hypothesized that these genes will be able to make compounds with structures similar to the alpha-letoic acid we're looking for. We clone these enzymes, input them into a heavily engineered and generally regarded as safe fungal host, and we were able to produce new compounds shown here, these 4 compounds. And it turns out one of the compound 3, turned out to be alpha-letoic acid. And with very little optimization, we're able to achieve about 80 milligrams per liter of this compound. But a majority of the compound produced is a slightly different version of alpha-letoic acid, also known as fluorocarboxylic acid. And this compound contains 2 additional carbons on the side chain compared to alpha-letoic acid, and it's produced at an astounding 1.4 milligrams per liter level. So well exceeding any known levels of compound production based on previous efforts. This compound, this longer compound can be, if we can elaborate that into the final cannabinoid molecule in THCP, it could be quite significant because it's been studied that THCP compared to THC, which contains 2 less carbons, this new molecule is a rare cannabinoid, and it's the most potent cannabinoid inhibitor known with single-digit nanomolar affinity for the cannabinoid receptor. But it's very difficult to access this compound because it's a rare cannabinoid. So we think that with our platform, being able to access this unusual intermediate, we'll have a very direct way of obtaining this potent inhibitor. We also test the other pathway. We can show that one of the pathways that we found can produce cannabinoid as a single, the alpha-letoic acid as a single compound, hence allowing us to make this compound take this pathway further into the actual cannabinoids. So let me just summarize here that our challenge low titer, we have demonstrated new pathways discovered in fungi can produce key intermediates at gram per liter scale, which is a significant first step. We have identified a completely new pathway and have filed for new IP from UCLA. And in terms of diversification, we think that this 2 polyketide system in these new fungal pathways offer unique routes for structure diversification. So what will we do in this proposal? Together with Professor Garg, we will take advantage of both enzyme chemistry and synthetic chemistry to make variants of alpha-lipoic acid. For example, we'll perform additional genome mining to find other pathways that can produce related compounds. We will engineer the proteins to produce structurally diverse precursors that can be fed into the second polyketide synthase. We will synthesize chemical precursors and directly feed into the second enzyme to access different versions of this 2,4-dihydroxybenzoic acid. And then we will optimize the host using synthetic biology efforts. And lastly, in parallel, we will extend this pathway into the actual THC and CBD and their derivatives. We will do so by taking advantage of this synthetic biology infrastructure that have been recently installed and up and running in the second floor of CNSI. This is the Biopacific, part of Biopacific, and this is headed by Mike Lake, the staff scientist, the position, and two of my students, Ike Okorafor and Elizabeth Abramian, are setting up first set of experiments using this high throughput synthetic biology robotic system to make variants of our pathway to make new cannabinoids. Lastly, I want to thank the Nobel Fund for very generous support and also thank CNSI, Biopacific, NSF, and NIH for allowing us to obtain some of these preliminary data. Thank you very much. I'm happy to take any questions.